Okay, in this video I'm going to show you cellular respiration. So let me orient you to this picture so far. So this entire screen would represent a cell. The cell would be much bigger actually than this screen. And then this right here is our mitochondrion. So this would be like cytoplasm in and around here. And this is our cell membrane. And then this is a capillary that's in close contact with the cell membrane. Most cells, all cells, are in close contact with some kind of capillary or blood source for their nutrients. Okay, so let's say it's first thing in the morning and we've just woken up and our cells are hungry and we're hungry, and so we go ahead and we eat some Lucky Charms, let's say. And so now in our bloodstream we've got all these little glucose molecules floating around, and they want to get inside the cell because it's high concentration in the cells and are in the capillaries and low concentration in the cells. And so in order to do that, we have this really cool protein called GLUT4. You don't need to remember this, but this is just kind of an FYI. It's kind of a fun protein. It's the protein that responds to insulin. And so if you don't have insulin, this channel protein won't be in the membrane and glucose cannot get in the cell. But if you have insulin, the GLUT4 comes to the membrane surface and acts as a channel. And so what you'll have happen is, because it's high concentration in the capillary and low concentration in the cells, the glucose will begin to flow through the channels into the cells, which is, if you remember, facilitated diffusion. And so now I'm going to draw the glucose in the cell, and I'm just going to make it a 6-carbon sugar. I'm going to make it a chain instead of a circle. That's one of the breaks that happen. We're not going to worry about that. So here we have this chain, and to get the energy out of it, we have to break it apart. So we are going to snap a bond, right? And when we snap that bond, we're going to end up with, obviously, 2 3-carbon sugars. And they have a name. They're called pyruvate. Okay, and in order to break those, we have to put a tiny bit of energy in, because if you remember, all reactions need energy of activation. So we're going to put a tiny bit of ATP in to get the bonds weak enough to break. But you're going to get more energy out. So remember, from the activity we learned before, when you break a bond, you're going to get 2 high energy electrons. Okay, we're just going to draw them like this, really high energy electrons. Okay, and then you also get a tiny bit of extra energy out, so we actually get 4 ATP out for the 2 ATP we put in, so it makes energy. Now, do you remember what we do with these electrons in order to keep them at high energy and not let them fall down? Well, we put them right back into a bond between NAD and H, so we're going to store those 2 electrons. And this whole process that we've just drawn is called glycolysis, which quite literally means sugar, glyco, breaking. Glycolysis. To get the S on there. Okay, and so this happens in the cytoplasm. So you can see that anybody can do this. They don't have to have a mitochondria. So bacteria can do this because it all happens in the cytoplasm. So that's step 1. Okay, now if you remember from the activity, we have to continue breaking down these bonds. But the rest of this process happens inside of the mitochondria. And so we're going to take our 3-carbon sugars into the mitochondrion, into the matrix. I'm just going to draw my 3-carbon sugar again. And we're going to continue breaking it down. So the next step that happens is we break a bond. Now we have this single carbon atom, which is going to leave as CO2, carbon dioxide. And that's just going to transfuse out of the membrane into our blood cells and be returned to the heart that we exhale. So notice the very first exhale, we're exhaling sugar. So the Lucky Charms you ate this morning gets exhaled as the air you breathe out later in the day. Pretty crazy, right? So then we have these 2 carbons still, and they're going to break down. This little step here doesn't really have a name. Let's go back to it though. Since we've broken bonds, we've actually released electrons, right? And we're going to store them in our NADH. Okay, we don't get any extra energy out, so that's it. And then we need a little helper. It's a coenzyme. So we call these helpers. We call it coenzyme A. It's a little helper that's going to take this little 2-carbon sugar, which we call an acetyl, over to the next step. For whatever reason, it needs a little chaperone to get to the next step of the process, which is the citric acid cycle, which I'm going to abbreviate citric acid cycle. And it's a cycle because it starts and ends with the same product. It actually starts and ends with a 4-carbon sugar. That's 4. And we're just going to add the 2 carbons to it. And then we're going to kick those 2 carbons out when we break that last bond. And they're going to leave as CO2. doing the same thing, diffusing out of the cell. Okay, now we've just broken another bond, so what have we done? Well, we've taken electrons out, and we're going to store them. And we happen to get a little bit of extra energy out of this process. We get 1 ATP per turn of the cycle. The cycle is going to turn twice, right? Because we've got 2 original pyruvates that have to go through this cycle. So everything inside of the mitochondria happens twice. Okay, so now if you count, we've lost all 6 carbons. They've gone and they've been exhaled as CO2. So all we have left now are these high energy electrons stored as NADH. We got some here, we got some here, and we got some here. So now we have to take those electrons down one step at a time. So do you remember this process that we talked about in our first activity where we're taking electrons from really outer layers down to lower layers? And it turns out these proteins that I've got drawn right here in the membrane, these each have a protein or have an electron shell, one lower than the one before them. And so our electrons are bouncing from protein to protein and losing energy as they go. Okay? And so what we're doing is we're taking all the electrons from all of our NADHs and we're dropping them off. So that my red color is representing electrons. These are all the electrons that are high energy, and they're going to bounce from one protein to another. And as they do, they're releasing a little bit of energy. And what are we going to use that energy to do? Well, we're going to use it to shuttle a hydrogen ion across the membrane to here. Okay, I'm going to draw them as little dots because it's too hard to draw little H's. So we end up shuttling all these hydrogen ions to this side of the membrane every time the electron drops in energy. Okay, the very last drop that the electron makes is onto an oxygen. Because oxygen is so electronegative, it's constantly stealing the electrons from this guy, who then steals them from this guy, who steals them from this guy, who steals them from NADH. So as long as oxygen is present, it will pull electrons down and this process will occur. Where did the oxygen come from? Well, it was hanging out on the red blood cells, right, when you breathe in, and it'll simply just diffuse across the membrane and make its way to free electrons because it's so electronegative. And when you add electrons to oxygen, it turns into water because it has negative charge and it attracts those positive hydrogens. So the air you breathe in turns into water, not the carbon dioxide you breathe out. The air you breathe in turns into water. The carbon dioxide comes from the sugar. Now the very last step we have is all of these protons, hydrogen ions that have been pushed to this side of the membrane, they all want to go back out. So they're going to flow through our ATP synthase, and that's going to pump out ATP by spinning. You remember that? Okay, so we've produced ATP in a couple places. We've produced it here, we produced it here, and we produced it here. Now here was only like 2, here was 1 per cycle. This produces 32 to 34 per cycle. So this is a way more efficient way of making ATP, and it's called oxidative phosphorylation because it requires an oxygen. Phosphorylation just means to add that third P to ATP. Whereas these 2 ATPs right here, these were created through what we call substrate level phosphorylation. That's just where we grabbed it off one thing, put it on another. It's nothing fancy. Oxidative phosphorylation is like super fancy. We're taking oxygen, we're pulling hydrogen ions across a membrane, we're shoving them back through ATP synthase. So that is cellular respiration. You're going to draw it for yourselves, and then you're going to explain it to 5 people. Good luck.